Good evening, everybody. Welcome to this evening episode of Pursue. This is Pursue 16F, which is Hematology Non-Neoplastic Lesions of WBC. We are streaming live from the famous iconic IPGMER, Kolkata, India. And we have once again Dr. Shristi Butta with the session 4 of this. Today she'll be dealing with the structure of lymphocytes, plasma cells, lymphopoiesis, immunoglobin production, T cell receptors. Dr. Shristi Butta is an MBBS honors, gold medalist, MD pathology honors. Presently, she is a demonstrator in the famous IPGMER SSKM Hospital, Kolkata, with areas of interest in cytopathology, hematopathology, and oncopathology. Before I ask her to start, let me request all of you to keep your mic muted, your camera off, and please don't share your screen. With this, let me request, ma'am, please share your screen. Let's start.
Hello.
Hello?
Hello. Sir, is my screen visible?
Yeah, slightly increase the size of the screen.
So now?
Yeah, that's fine. Perfect. Please start. Thank you.
Yes. So, a very good evening to all of you. Today's session is on the non-neoplastic disorders of WBC session 4, and this is about lymphocytes and plasma cells this time. So, the overview of the seminar includes lymphocytes, T cells, B cells, and NK cells, the structure, development, functions of lymphocytes, lymphocytosis, lymphopenia. Special mention will be given to COVID-induced lymphopenia, NK cells and associated disorders, plasma cells and associated non-neoplastic disorders, as well as tumor-infiltrating lymphocytes, and there will be some key takeaways. So let's begin. Now, lymphocytes. Lymphocytes are responsible for an astonishing specificity of adaptive immune responses. Lymphocytes develop in the thymus and bone marrow, which are therefore called the central or the primary lymphoid organs. The newly formed lymphocytes migrate from these primary lymphoid organs to the secondary lymphoid organs. There are about 2 into 10 to the power 12 lymphocytes in the human body, making the immune system comparable in cell mass to that of the liver or brain. As mentioned earlier, lymphocytes usually respond to foreign antigens only if the innate immune system is first activated. So let's see how does that happen. We already know that microbes enter through skin or okay, and there onwards what happens is that the dendritic cell matures and carries these microbial antigens to local lymph nodes to become antigen presenting cells. These antigen presenting cells activate T cells to respond to microbial antigens. Activated T cells thereafter migrate to the site and thereafter eliminate these residual microbes. Now, coming to the development of lymphocytes, how do these lymphocytes develop? Within the bone marrow, we have these hematopoietic stem cells, right? So these hematopoietic stem cells convert into common lymphoid progenitors, that is, the common lymphoid progenitors. These common lymphoid progenitors subsequently change into the early thymic progenitors. These early thymic progenitors change into the double negative progenitors type 2 and type 3. We will further go into the details of it in the subsequent slides. This further on changes into the double positive progenitors and subsequently the single positive progenitors. Now let's see what else. Hematopoietic stem cells differentiate into multipotent progenitors. These multipotent progenitors initiate the transcription of the recombination activating genes, that is the gene 1 and gene 2, and become the lymphoid primed multipotent progenitors, and then the common lymphoid progenitors, as we already discussed. A subset of the pluripotent cells migrate to the thymus and differentiate into the early thymic progenitors. Now, what does happen in the thymus then? In the stroma of the thymus, the early thymic progenitors encounter a large number of ligands called as the Notch receptors. The function of these Notch receptors, that is the Notch1 receptor, and their interaction with the delta-like ligand. So remember that you have the Notch-like receptor, okay, which interacts with the delta-type ligand, right? So what has happened? The expression of Notch1 receptors and the interaction with the delta-like ligand is essential for the differentiation of the T cells in thymus and for the inhibition of the non-T cell lineage. So it allows for the maturation of the T cells and inhibits the non-T cell lineage. Within the thymic cortex, we have the early thymic progenitors which differentiate further on into the double negative cells. It should be noted that the early thymic progenitors are the double negative type 1 cells, that is the DN1 cells. The early thymic progenitors later on differentiate into the double negative type 2 cells when it acquires CD25 and CD44. At this stage of development, the cells lose their B cell potential and start expressing what is called as the RAG1 and the RAG2. DN3, that is the double negative 3 cells, can take two divergent routes. Now, what are the two divergent routes? That is the alpha beta chain and the gamma delta chain. Let's go into it. What is the difference between the alpha beta T cells and the gamma delta T cells then? The alpha beta T cells recognize antigens or peptides only when they are coupled with what is called as the MHC or the HLA. The MHC will be class 2 in case of CD4 plus T cells and class 1 in case of CD8 plus T cells. Gamma delta T cells, on the other hand, recognize antigens, that is proteins and non-proteins. So they are the ones which are both CD4 and CD8 negative. That is the DN type, double negative type. Okay. So it is important to note that these are nothing but the intestinal intraepithelial lymphocytes. They are present predominantly in the gut. It is further on important to note that the alpha beta T cells are the ones which release cytokines as well as behave as cytotoxic T cells. They are involved in immune protection and pathogen eradication. On the other hand, we have gamma delta T cells which are involved predominantly in immunoregulation and immune surveillance. Right? So it is particularly very important to note that the alpha beta T cells are the ones which are involved in immune response. On the other hand, the gamma delta T cells are particularly involved in the immune regulation. The expression of the beta chain of the T cell receptor at the DN3 stage, remember the DN3 stage, there the alpha beta T cell receptor is changing, and we also have the gamma delta T cell receptors there. The DN3 cascade then simultaneously allows for the expression of CD4 and CD8 cells. So in that case, that results in the double positive progenitor development. On the other hand, the double positive thymocyte further on differentiate into the single positive thymocytes. These single positive progenitors are either CD8 positive or they are CD4 positive. That is, they either respond to HLA class 1 or HLA class 2. Double positive cells incapable of binding to HLA1 or HLA2 undergo what is called as apoptosis. So if you have the double positive cells that are incapable of binding to HLA1 or HLA2, then they undergo apoptosis. T cells subsequently leave the thymus and migrate continuously to the secondary lymphoid organs to be primed and differentiate into effector cells with specialized phenotypes. Now, coming to the T cell receptor complex, the T cells acquire a receptor called as the T cell receptor that recognizes specific antigens. The T cell receptor is a multipotent complex. What does it include? It includes the alpha beta or the gamma delta T cells. Okay, the alpha beta T cell receptor does not recognize antigen in its natural form. It recognizes antigens only when it is coupled with either HLA1 or HLA2. The peptides presented by HLA1 molecules are small and have an intracellular origin. They are small, having an amino acid length of about 8 to 10 amino acids. On the other hand, those that are presented by HLA class 2 are usually 13 to 25 amino acids in length and are generally extracellular in origin. Let's move on further. We have the T cell receptor, the alpha chain, and we also have the beta chain. Okay, so the alpha chain of the T cell receptor basically is presented on chromosome 14, whereas chromosome 7 encodes the beta chain of the T cell receptor. It is important to note, just look at the diagram, each chain has a variable region, that is a V, a constant region, that is a C, and the joining segment. The beta chain also has an additional diversity segment or the D segment. Each B domain has 3 hypervariable sectors, that is the CDR 1, 2, and 3, here 1, 2, and 3. Okay, so what is this? This is nothing but the complementary determining region, and that is capable of generating an immense pool of combinations that particularly react to different antigens in different manner. So it is nothing but the CDR or the complementary determining region which allows for the formation of antigenic specificity for the T cells. Now, coming to the different types of T cells, yes, we all know it is the CD4 plus T cells or the naive T cells. Now, what happens is that they on dividing into T helper 1 cells, 2, 9, 17, 22, and the follicular T helper cells, right? So what happens is that the T helper 1 cell is capable of producing interferon gamma. The interferon gamma promotes cellular immunity against intracellular pathogens and participates in what is referred to as autoimmune disease pathogenesis and delayed hypersensitivity reactions. T helper 2, on the other hand, produces interleukin 4, 5, 9, 13, 10, and 25. It promotes for IgE class switching, mast cell recruitment, eosinophilia, mucus production. It is further on very important to note that T helper 9 cell allows for promoting IgE class switching, mast cell recruitment, and mucus production as well. T helper 17, on the other hand, produces cytokines that are responsible for neutrophilia. Yes, remember the class on neutrophils that we discussed? We always discuss these T helper 17 cells, which is responsible for the neutrophil recruitment. It is very important to note further on that we have T helper 22, that is involved in antimicrobial peptide production, and it also promotes keratinocyte proliferation and inhibits their differentiation. Follicular T helper cells, on the other hand, induces formation of germinal centers, transformation of B cells into plasma cells, and production of antibodies with different isotypes and production of early B cells. So this T follicular cells will be discussed in just a bit. Now, what happens is that you also have the CD8 plus T cells. Now, what are these? These are the cytotoxic T cells, as we all know, right? Now, these CD8 plus T cells develop its effector function by converting into the cytotoxic T cells. A cytotoxic T cell induces apoptosis in target cells by liberation of the cytolytic granules or by expression of the ligands for the death receptors or the FAS-L. Okay, remember the pathway of apoptosis? Now, perforins polymerize in the plasma membrane and produce pores which act as channels that allow water entry and generate an osmotic disequilibrium. Okay, so what is very important is that these perforins are going to cause osmotic disequilibrium by producing pores which act as channels. Perforins also facilitate the passage of granzymes to the cytosol and to the nucleus of the target cell. So very, very important is that the perforins are facilitating the passage of these granzymes to the cytosol and the nucleus, which favors the proteolytic action on the mitochondria as well as fragmentation of the DNA. These cytotoxic T cells also liberate interferon gamma and TNF alpha, which are important in the defense against viral infections and in controlling the proliferation of tumor cells. So besides the antiviral action that it has, it also is cytotoxic to the tumor cells. Now, coming to the B cells or the B lymphocytes, it is important to note that these lymphocytes develop within the bone marrow from the hematopoietic stem cells, and so these stromal cells of the bone marrow act as a MIKEY for these B cells. Interleukin 7 is very critical because it aids in the development and production of the B cells. It is produced by the stromal cells of the bone marrow. Moving on to the development of the B cells, we have the hematopoietic stem cell here that further on converts into the early lymphoid progenitors that is capable of converting to the common lymphoid progenitor. The common lymphoid progenitor type 2 thereon converts into the early pro-B cells, late pro-B, pre-B and immature B cells. These immature B cells express the membrane-bound immunoglobulin. Okay, it moves on into the spleen and undergoes class switching and produces different types of antibodies depending on the stimulation, and also produces what is called as the short-lived plasma cell. This thereon moves on to the long-lived plasma cell. Let's see does that happen. The B cell receptors. Now, during the differentiation of B cells, a process of gene recombination is structured initially that codes for segments, that is the variable, diversity, and the joining of the heavy chain. Similar to that is that of the light chain, where we have the variable and the joining segments of the light chain, bound to the membrane-bound immunoglobulin. This recombination process is initiated by the protein, that is the RAG1 and RAG2. That generate the rupture of the double chain of DNA between the segments of genes and subsequently allows for expression of the recombination signal sequence. This process leads to the generation of B cells that expresses a wide repertoire of membrane-bound immunoglobulin and produces what is characteristically called as the B cell receptor. And it is this B cell receptor which is responsible for the antigenic variability. But how does that happen? Stimulation of the B cells via the antigenic B cell receptor begins with the recognition and capture of the antigen through the B cell receptor molecule. This induces their aggregation and triggers the signaling process by activating the SRC family kinase. It is important to note that this SRC family kinase is the tyrosine kinase. The activation of the tyrosine kinase pathway allows for cell survival, proliferation, and migration. So it must be noted that this tyrosine kinase pathway, that is the RAS and the PI3K-AKT-mTOR pathway involving NF-kappa B, is also particularly very important for the synthesis of the B cells. Now, this is what is called as the signalosome. Now, this complex that we are trying to depict here is nothing but a signalosome. Now, this initiates the formation of a macromolecular complex, which is called as the signalosome, composed of the B cell receptor. You have the tyrosine kinases. We also have the CD19 and the BLINK, that is the B cell linker protein. Signaling enzymes such as the phospholipase C, gamma phosphatidyl inositol 3 kinase, that is the PI3K AKT mTOR pathway activation, and the signaling produced by this signalosome further on activates multiple signaling cascades and initiates what is called as the NF-kappa B, BCL6, NF-AT, FOXO, June, and ATF2, etc. So what is particularly very important is that this is involved in cell proliferation, migration, and survival of the B cells. Now, B cells are activated when their B cell receptor binds to either soluble or membrane-bound antigen. This activates the B cells to form microclusters, and that triggers a downstream signaling cascade. The microcluster eventually undergoes a contraction phase and forms an immunological synapse, and that allows for what is called as the class switching. So once activated, let's see what happens. Mature B cells express IgM and IgD as their first heavy chain segments in the immunoglobulin locus, right? After a B cell is activated, the segments of the locus are cut. So here it is cut here and it is cut here. Okay, and thereafter it is joined. So that allows for what is called as the class switching, and now another heavy chain segment, that is the IgE, is first on the immunoglobulin locus, and that is what is expressed here. There are 5 different types of immunoglobulins, as we all know. It is the IgG, A, M, E, and D. It is well known now that B cells play a fundamental role in the pathogenesis of several diseases, particularly the rheumatoid arthritis, or for that matter, SLE, and even multiple myeloma. The production of autoantibodies and its secretion of chemokines and cytokines, and there may further on assist in antigen presentation. Thus, there are several receptors and co-stimulatory molecules in parts of the signaling cascade that can be targeted for treatment planning in rheumatoid arthritis, SLE, and multiple myeloma patients. So after the B cells, now we have the plasma cells. Plasma cells are differentiated B cells. Okay, so what do they do? They are capable of secreting a lot of immunoglobulins or antibodies, and these cells play a significant role in what is called as the adaptive immune response. Now, plasma cells develop from antigen-activated B lymphocytes in secondary lymphoid organs. Remember the secondary lymphoid organs that we talked about, such as the spleen and the lymph nodes. Okay, and the important transcription factors that are involved in the differentiation of the plasma cells from the mature B cells are LIM1, IRF4, and XBP1. Plasma cells are nothing but the antibody factories. The most immature blood cell of a plasma cell lineage is a plasmablast. The plasmablast can proliferate and secrete small amounts of antibodies. The terminally differentiated or mature plasma cells are non-proliferating and are much larger than B cells and can secrete larger amounts of antibodies. They have a short lifespan, that is about 2 to 3 days. They continuously synthesize and secrete antibodies with high specificity for antigens that stimulate the stimulated plasma cell precursor to proliferate and differentiate. Though the production and secretion of antibodies were long thought to be the sole function of plasma cells, now we know that they are also involved in immune response regulation. Within this capacity, research has found that plasma cells to inhibit the development of follicular T helper cells. Remember the follicular T helper cells that we talked about in the last few slides? It has been found in several researches that mice devoid of plasma cells, via a knockout of the BLIM1 gene, expressed elevated levels of follicular T helper cells. Okay, so if you have a knockout of the BLIM1 gene, then that is resulting in a mice with devoid plasma cells, and that is allowing for an elevated levels of follicular T cells, which in turn produces interleukin 21, and that is a protein which is critical for the processes of affinity maturation, germinal central longevity and function, and B lymphocyte terminal differentiation. Right? Okay, so now we have the histochemistry. All plasma cells express CD38 and 138. So this is a picture shown alongside. It's been taken from Pathology outlines, and it's a case of chronic endometriosis, okay, where you have abundant plasma cells forming clusters, and that's been highlighted by CD138. CD38 catalyzes the synthesis and hydrolysis of cyclic ADP ribose, resulting in the maintenance of the intracellular calcium levels. In contrast, CD138 is also called as the syndicam-1 and allows the plasma cells to attach to extracellular matrix proteins. Recent research has found that in addition to its role in plasma cell adhesion, CD138 plays a critical role even in acting as a co-receptor for the epithelial growth factor, particularly in the context of multiple myeloma. Though malignant plasma cells express CD38 and 138, it must be noted that alongside their normal or non-malignant counterpart, a distinguishing characteristic feature of a malignant it must be noted that this CD56 is a marker of the NK cells. However, it also bears a mention that not all myeloma cells express CD56. So besides CD38, 138, loss of CD19, there is also an expression of CD56, but however, not all plasma cells express CD56 in multiple myeloma. An additional noteworthy marker of plasma cells is CD79A. The protein plays a critical role in B lymphocyte antigen signal transduction and overall B lymphocyte development and stabilization. Though CD79A is present in cytoplasm of both the precursor B lymphocytes and mature lymphocytes, differentiated plasma cells, its apparent loss of expression has been noted in certain samples of plasma cell neoplasm. So its loss of expression is also one of the keys to the diagnosis of a plasma cell dyscrasia. Coming to the light microscopy, plasma cells vary in size from 14 to 20 micrometer. They are about 14 to 20 micrometer in size, round to ovoid cells with a eccentric nucleus, yes, and a pale perinuclear area, which is nothing but the Golgi apparatus. They are round, and it is further on important to note that there are several other forms, like the Mott cells. We also have the Russell bodies here. Okay, the Mott cells and the Russell bodies are nothing but the dilated endoplasmic reticulum cistern containing condensed immunoglobulins, right? Coming to the electron microscopy reveals that plasma cells contain an eccentric round nucleus with condensed chromatin and a well-developed nucleolus. The nuclear membrane associated chromatin gives the nuclei of mature plasma cells what is called as the cartwheel appearance on histological sections. The cytoplasm contains parallel arrays of rough endoplasmic reticulum, cytoplasmic fibrils, and numerous polymorphic mitochondria. The cytoplasm also contains Golgi apparatus, yes, and that causes what is called as the perinuclear Hoff. Now coming to the non-neoplastic disorders of plasma cells. Let's discuss that first. We have common variable immunodeficiency. Okay, now this common variable immunodeficiency presents in patients who have an IgG level that is less than 400 milligram per DL. Okay, however, if IgG levels decrease to beyond less than 200 milligrams per DL, then that results in what is called as a severe common variable immunodeficiency, and that particularly requires what is called as replacement therapy. The lack of immunoglobulin, which is very important, results in an increasing susceptibility to bacterial infections. Okay, patients usually present with abdominal pain, chronic or bloody diarrhea, organomegaly, alopecia, lymphadenopathy, bronchiectasis, cough, dyspnea, lung infiltrates, and pleuritic chest pains. Skin manifestations like actinic keratosis may also be seen with hyperpigmentation in petechiae. Systemic manifestations of malnutrition and weight loss is also commonly seen. Such patients usually are affected by Haemophilus influenzae, varicella zoster, pneumococcus, etc. Now, coming to X-linked A-gamma globinemia, yes, we all know that this is a condition which basically results due to mutation in the BTK gene, okay, or the Bruton's tyrosine kinase gene. Now, that is particularly involved in converting the pro-B cells to the pre-B cells. So, any defect results in the lack of formation of the B cells and subsequently A-gamma globinemia. Because it's an X-linked disorder, males are exclusively affected. Coming to the selective IgA deficiency, the most common primary immunodeficiency affecting the males and females equally, as implied by the name, there's a selective IgA deficiency. Okay, so IgA deficiency particularly results when there is either an improper isotype switching to IgA or an improper IgA secreting B cell maturation. Selective IgA deficiency, usually patients are asymptomatic or have airway and GI infections, autoimmune diseases, atopy, anaphylaxis to IgA containing blood products. Now, coming to the causes of lymphocytosis, first the definition. Lymphocytosis is defined as an increase in the absolute lymphocyte count, that is about more than 4,000 lymphocytes per microliter in adult patients. Lymphocytes represent around 20 to 40% of the total WBCs. The definition of a relative lymphocytosis is an increase in the WBC, that is more than 40%, in the presence of an absolutely increased WBC count. So we have reactive or atypical lymphocytes. They typically have what is called as skirting here. That is what you see. We have infections like viral infections, EpsteinBarr virus, cytomegalovirus, HIV, herpes, influenza, varicella, HTLV, bacterial infections like pertussis, parasitic infections like toxoplasmosis. We also have autoimmune conditions, hypersensitivity reactions, and hyperthyroidism, trauma, post-splenectomy patients, and even cigarette smoking can cause reactive lymphocytosis. Now, coming to that one by one, we have the Epstein-Barr virus that is particularly involved in what's called as infectious mononucleosis. Patients usually present with fever, fatigue, sore throat, and have swollen lymph nodes. Yes, so that's a very common viral infection that predisposes patients to lymphocytosis. And another important thing is that it must be noted that it is the lymphadenopathy, pharyngitis, splenomegaly, and various hematological manifestations may be seen in this condition. Cytomegalovirus again can cause a disease which is indistinguishable from Epstein-Barr virus induced infectious mononucleosis. We also have HIV infection where there is a lymphopenia. In addition, it must be noted that though chronic HIV infection can cause lymphopenia, a decreased CD4 plus T cell count in primary HIV infection can cause an acute febrile mononucleosis like illness and predispose to what is called as lymphocytosis. Okay, a negative heterophile test can help in distinguishing the two. Other viruses like influenza, hepatitis, mumps, measles, rubella, etc. can also cause lymphocytosis. Some bacterial infections, besides causing neutrophilia, can exceptionally cause lymphocytosis, like Bartonella Hensley. Bartonella Hensley leads to a cat scratch disease, a disease transmitted by scratch or bite of a cat, and it is usually associated with ocular and neurological manifestations besides causing lymphocytosis, which is usually large and atypical lymphocytes are seen. In Bordetella pertussis, yes, there's the picture of pertussis in a child. It is characterized by increased lymphocytes that are usually small and have a deeply cleaved nucleus. Okay, let's look at the picture. Okay. We also have parasitic infections like Toxoplasma gondii. Symptomatic toxoplasmosis can also cause lymphocytosis. babesiosis, and even mycobacterial tuberculosis. So that you must not ever forget. Drugs and drug-related conditions. We also have the DRESS syndrome, that is the drug hypersensitivity reactions associated with drug reactions and eosinophilia and systemic manifestations can also lead to lymphocytosis. A relatively new CLL medication, that is the Bruton's tyrosine kinase inhibitor, ibrutinib, has also resulted in lymphocytosis. So it must be noted that it likely represents a redistribution of the CLL cells from the lymphoid tissue to the peripheral blood, and that results in lymphocytosis. So there is a redistribution of the CLL cells, and that causes lymphocytosis on giving ibrutinib. So that is basically a relative lymphocytosis. Monoclonal B lymphocytosis. This refers to the presence of monoclonal B cells at a level that is less than 5,000 cells per microliter in the absence of features of lymphoproliferative disorder like splenomegaly and or cytopenias. The disease may either be persistent or undergo a regression or even may progress to CLL. Okay, so monoclonal B lymphocytosis can progress to CLL. Congenital B lymphocytosis. Now, this is due to a germline heterozygous missense mutation of the CARD11 gene, a scaffolding protein which is particularly involved in the NF-kappa B synthesis. Now, it is important to note that NF-kappa B is particularly very important because remember the tyrosine kinase pathway and the cell proliferation and migration and survival that I was discussing, where it is important to note that these are particularly involved in the maturation of both the B lymphocytes as well as the T lymphocytes. So a congenital B lymphocytosis resulting in a germline mutation, particularly due to defect in the NF-kappa B. It typically progresses to CLL by the 4th decade of life. Persistent B-cell polyclonal B lymphocytosis. Now, this is a rare entity described as polyclonal binucleated lymphocytes occurring predominantly in young smoker women. Unlike CLL or monoclonal B lymphocytosis, lymphocytes are polyclonal here. Okay, it is associated with HLA-DR7 and IgM polyclonal gammopathy and exhibits a stable clinical and biological course. Stress, severe and emergency medical condition may also result in a transient lymphocytosis. Besides that, asplenia, similar to other blood component, lymphocyte count may also increase post-splenectomy. Now, the pathophysiology behind a rise in the lymphocyte count. Increased lymphocyte production may either be due to a clonal process or a reactive process. Yes, also redistribution of the lymphoid cells, okay, as seen due to CLL drug, that is the ibrutinib BTK inhibitor, which allows for the redistribution of the lymphoid cells, and that also results in lymphocytosis. Epstein-Barr virus. During the early stages of infectious mononucleosis, Epstein-Barr virus infects resting B cells, and a large number of infected B cells circulate in the blood. The atypical lymphocytes, however, are activated cytotoxic CD8 plus T cells that appear 1 to 3 weeks after the onset of symptoms. In the patients infected with Epstein-Barr virus, memory B cells are latently infected and leads to chronic infection with possible reactivation and predisposition to lymphoproliferative disorders. In pertussis, the mechanism of lymphocytosis is due to a decreased extravasation of lymphocytes from the peripheral blood to the lymphoid organs or expansion of morphologically normal CD4 plus T cells. Congenital B lymphocytosis, as discussed, is basically due to the mutation in the CARD11 gene. Another important aspect is the approach to the diagnosis of lymphocytosis. History and physical examination are particularly very important in such a case. Examination of the peripheral blood smear is particularly very important. It is important to note that these reactive lymphocytes are either present at the feathery edges, okay, or they tend to collect at the edge of the smear. A good practice is therefore to examine these lymphocytes. Yes, it is not uncommon for fragile leukocytes in CLL or even in infectious mononucleases or acute leukemia for that matter to smudge on blood smears. So how do we prevent that? In these situations, a few drops of albumin can be added to the peripheral blood smear, and these albumin smears allow for proper identification of leukocytes and reduce the number of smudge cells or basket cells. However, examination of RBCs and platelets should be performed on the original blood smear because albumin can affect platelet and erythrocyte morphology. So that is again very important. Reactive lymphocytosis, separating a monomorphic lymphocytosis from a pleomorphic lymphocytosis, can help distinguish a lymphoproliferative disorder from reactive lymphocytosis. Most reactive lymphocytosis show a wide range of size and shape. Okay, so the classic example of a pleomorphic lymphocytosis is infectious mononucleosis. In this spectrum of morphology that points to a greater likelihood that patient has reactive lymphocytosis, younger age is also very helpful clue, because usually patients with young age are affected by viral and bacterial infections. Autoimmune disorders, yes, there might be a history of vaccination, drug hypersensitivity, endocrine disorders, stress, smoking, and even a history of malignancy. While most of these reactive lymphocytoses are pleomorphic, very few important exceptions do exist. Okay, so you have Bordetella pertussis. The lymphocytes of Bordetella pertussis are small and deeply clefted, as this is commonly seen in the pediatric and pregnant population. So it is very important to differentiate it from the follicular lymphoma or from the Sézary syndrome. Let's see the slide. Here is a case taken from the American Society of Hematology. Now this is, these are the cleft-like lymphocytes, okay? So although like simulating the Sézary cells, right? But however, it was found to be a case of pertussis. The second exception is a polyclonal B lymphocytosis, which typically shows lymphocytes with distinct nuclear clefts, but will demonstrate a spectrum of morphological changes, including nuclear lobation and binucleated forms. So it is the polyclonal B lymphocytosis in that case. This uncommon disorder is found particularly in young and middle-aged female smokers with high association with HLA-DR7 and several genetic abnormalities. The final exception is a large granular lymphocyte. Okay, so this large granular lymphocyte can also be seen in viral infections, and so it needs to be differentiated from a large granular lymphocytic leukemia. Obviously, history would help in that. Now, this is, in short, a description of the lymphocytes seen in CLL, the small round nuclei, the follicular lymphoma and mantle cell lymphomas for that matter, or even the T cell lymphomas where you have the folded or the cleaved nuclei. Sézary syndrome, yes, you have the Sézary cell there with convoluted nuclei. The Willis cytoplasm of hairy cell leukemia, the plasmacytoid lymphoplasmacytic lymphomas and plasma cell dyscrasias. We also have granules within the cytoplasm in large granulocytic leukemia lymphomas. We also have prominent nuclei here in TPLL and vacuoles in the cytoplasm in Burkitt lymphoma. Now coming to lymphocytopenia. The term lymphocytopenia refers to a count that is approximately less than 1,000 lymphocytes per microliter in blood in adults. Okay. However, it may be less than 3,000 when talking about in children. Infectious diseases like AIDS, viral hepatitis, tuberculosis, and typhoid fever can cause lymphocytopenias. As acquired causes, we also have autoimmune disorders like lupus, steroid therapy, cancers like Hodgkin's and aplastic anemia, radiation and chemotherapy. Inherited causes include certain inherited diseases like Dye-George anomaly, Wiskott-Aldrich syndrome, severe combined immunodeficiency, ataxia, telangiectasia, and these inherited conditions are quite rare. Now, moving on swiftly to what is called as NK cells. Now, these natural killer cells constitute a very minor subset of the normal lymphocytes, and they typically account for about 10% of all the lymphocytes in the human peripheral blood. and provide an important effector in the arm of the innate immune system. Although named after their capacity to mediate spontaneous natural cytotoxicity, that is the natural killer cells, NK cells also are a major source of type 1 cytokines like interferon gamma as well as tumor necrosis factor alpha and GM-CSF. So what are these natural killer cells? Recently, NK cells were officially classified as the prototypical members of the group 1 innate lymphoid cells. Okay, so they were classified as the group 1. Note, it is important to note that they are the group 1 innate lymphoid cells. Group 2 innate lymphoid cells express interleukin 13, and it is important to note that group 3 expresses 17 and or 22. However, interleukin 13, 17 or 22 are not expressed by group 1 innate lymphoid cells, which characteristically expresses interferon gamma. The human NK cells are classically defined as CD56 positive but CD3 negative. It is important because it helps in distinguishing it from NK-like T cells, which are CD56 positive as well as CD3 positive. Two major subsets exist. One is the CD56 dim and the other is CD56 bright types. Now, CD56 dim NK cells are fully mature. They make up to 90% of the NK cells in the peripheral blood and predominantly mediate what is called as the cytotoxic response. CD56 bright cells are more immature, make about 5 to 15% of the total NK cell, and have been considered primarily as cytokine producers, and they have a limited role in the cytolytic or the cytotoxic responses. CD56 bright cells are better capable and are able to leave the vasculature and constitute majority of NK cells seen in the lymph nodes and even in the decidual tissues of pregnant women. NK cells can be rapidly activated to spontaneously attack certain abnormal cells in the body. Okay? Besides affecting the virus-infected cells, they also attack tumor cells. So that is particularly very important. Okay? Because NK cells, in that case, can be used in the treatment of carcinomas. Hence, rare individuals who selectively lack NK cells exhibit increased susceptibility to viral infections, herpes infections like herpes zoster, etc., and also an increased tumor incidence. So this figure shown alongside are the perforin-containing granules seen in NK cells. The predominant cytolytic target of NK cells are uncommon cells that have a downregulated MHC1. So it is very important that if you have a downregulated MHC1, then that in that case does not MHC1 loss is fairly common mechanism by which tumors and virus-infected cells can evade recognition by TCR of cytolytic T cells. NK cytotoxicity is mediated by directed exocytosis of cytolytic granules to release perforins and or granzymes which perforate the target cell membrane and trigger apoptosis. So it is particularly very important that cells which are deficient in MHC class 1 are the targets of NK cells. On the other hand, cells which have MHC class 1 are the targets for CD8 plus T cells, right? Or the cytotoxic T cells. So how do NK cells work? The discovery that NK cells selectively attack cells that have a diminished expression of MHC class 1 at first puzzled researchers in the early 1990s. The paradigm at that time had established that T cells and B lymphocytes rearrange genomic elements, okay, that is the RAG1 and RAG2, to create clonotypic antigen receptors. Therefore, it was unclear how NK cells could detect and respond to the loss of a normal ubiquitous protein like MHC class 1. But the myth was busted. This happened when mature NK cells were well tolerated towards normal cells by their expression of germline encoded inhibitory receptors that recognized MHC class 1. When these inhibitory receptors encountered MHC class 1 on normal cells, they recruited sonic hedgehog protein 1 and 2, tyrosine phosphatases at immunoreceptor tyrosine based inhibitory motive to dominantly arrest tyrosine kinase based activation signals. So, when a mature NK cell encounters a rare abnormal cell lacking MHC class 1, inhibitory receptors are not engaged, therefore, no recruitment of SHP1 or SHP2, and that causes an unsuppressed activating signals that triggers an attack on such cells which lack MHC class 1. So it was particularly important that these cells lacking MHC class 1 could not recruit SHP1 or SHP2, and that in turn led to their killing. Yes, it was busted. Now, classification of natural killer cell deficiencies. We have the classical natural killer cell deficiency. Okay, here it's a busy chart. Yes, you have the classical natural killer cell deficiency, which typically occurs when there is a deficiency of the NK cells. The NK cells are absent. That is, those cells which are CD56 positive but CD3 negative are absent here. Okay, that is the CD56 dim and the bright types usually. Okay, except the subtype 2, where the bright NK cell that accounts for about 5 to 15% of the NK cells is usually preserved. Such patients are usually susceptible to infections like herpes, varicella zoster, cytomegalovirus, mycobacterium, tuberculosis, trichophyton, etc. Functional NK cell deficiency particularly happens when these NK cells are present but they are functionally absent. So that happens when they are functionally defective. Infectious susceptibility involves herpes virus, herpes simplex, Epstein-Barr virus, human papilloma virus, etc. The genes that are classically defective in classical natural killer deficiency are basically the GATA2 and the MC-M4. Though the subtype 1 is autosomal dominant, the subtype 2 is autosomal recessive. Functional NK cell disorder particularly arises when there is a defect in the gene encoding FcGR3A. Okay, and it is important to note that this is an autosomal recessive disorder. Now, something very interesting is the CAR-engineered NK cells. Okay, the chimeric antigen receptors, engineered NK cells. Now, they are quite similar to the CAR-engineered T cells. Similar to the T cells, specific recognition and elimination of cancer cells can be undergone by even the NK cells, the chimeric activated NK cells. These chimeric antigen receptors, or the CAR NK cells, provide an opportunity to generate NK cell therapeutics of defined specificity for cancer immunotherapy. Let's see how. If you have an ex vivo activation with cytokines, can restore cytolytic activity of the NK cells against tumors, indicating that NK cells have a potential for adoptive immunotherapy if potent cytotoxicity can be maintained in vivo. Right? So if we can, in an ex vivo fashion, activate these NK cells by cytokines and then inoculate it in vivo, they can cause cytotoxicity to the tumor cells. So clinical trials are underway for many such tumors, including glioblastomas, melanomas, several metastatic tumors, rectal adenocarcinomas, etc. Lymphopenia predicts disease severity in COVID-19 patients. So in this era of COVID-19, okay, we have it all, right? Right from SARS-CoV-2 to now the present strain that we have in the Omicron, or the Omicron for that matter. Four potential mechanisms that lead to lymphocyte deficiency in COVID-19 typically happens when the virus might directly infect the lymphocytes, resulting in lymphocyte death. These lymphocytes express coronavirus receptors, that is the ACE2. It is important to know that. When these lymphocytes express coronavirus receptors, these are susceptible to attack by COVID-19, and that may be one of the reasons why lymphopenia occurs in patients with COVID-19. The virus might directly destroy the lymphatic organs. In addition, inflammatory cytokines like TNF alpha, interleukin 6, okay, IL-6, remember, and other pro-inflammatory cytokines can also lead to lymphocyte apoptosis. Inhibition of lymphocytes by metabolic molecules produced by metabolic disorders, okay, particularly metabolic acidosis or hyperlactic acidemia that happens in COVID-19 patient also inhibits lymphocytes. So lymphopenia is an effective and reliable indicator of severity and hospitalization in COVID-19 patients. So even lymphocytes can make such a big difference, right? So lymphopenia is effective and a reliable indicator of severity in COVID-19 patients. Now, just a brief overview, though we're talking about non-neoplastic disorders, just a brief overview about the tumor-infiltrating lymphocytes. Yes, this is a hot topic of discussion in a lot of journals and articles now. The TILs, or the tumor-infiltrating lymphocytes, are defined as lymphocytes that directly oppose and or surround these tumor cells. The degree of tumor-infiltrating lymphocytes may be defined by both the extent and the density of the tumor-infiltrating lymphocytes. The grading can be done, either it can be absent, or it can be a non-brisk response, or a brisk TIL infiltrate, which is defined as the presence of a diffuse permeation of the tumor by TILs or a tumor-infiltrating lymphocyte across the entire base of the tumor. So here you have, these are the tumor-infiltrating lymphocytes here, okay, and here you don't have any. Now, harvesting these TILs. Now, TILs are very important because TILs are cells that can be harvested from the tumor sites, and they can be expanded ex vivo, okay. So how does that happen? TILs, when expanded ex vivo, okay, with interleukin 2. So studies have demonstrated that melanoma TILs, that is the melanoma tumor-infiltrating lymphocytes, when cultured with interleukin 2, could consistently produce cytotoxic T cells. So TILs, when cultured with interleukin 2, could produce consistently cytotoxic T cells. Yes. So what could they do then? Yes, they could then lyse the tumor cells when given back to the patient. So similar results have also been found in glioblastomas and other metastatic carcinomas. So that is really very interesting, right? So an ex vivo expansion with in the presence of interleukin 2 can actually result in formation of cytotoxic T cells. So finally, we are coming to our key takeaways. Lymphocytes play a decisive role in maintaining immune homeostasis, inflammatory response throughout the body. Lymphocytes are white blood cells. Yes, they include the T, B, and the NK cells as the primary agents responsible for adaptive immunity. T cells and B cells sometimes are called as the special ops of the immune system. The CAR-engineered NK cells, the chimeric antigen receptor engineered NK cells, are similar to the CAR T cells. They are turning the innate effectors into more precision tools for cancer immunotherapy. Lymphocytes play a critical role in COVID-19 prognosis. Lymphopenia can be an effective and reliable indicator for severity in COVID-19 patients. Tumor-infiltrating lymphocytes can recognize and kill cancer cells. Yes, these lymphocytes, when cultured with interleukin 2, could convert into cytotoxic T cells. That can be used in cancer immunotherapy. And that opens newer avenues for future drugs in cancer immunotherapy as well. Thank you so much for your patient hearing.
Thank you so much, Dr. Shristi. As usual, an excellent superlative performance. My God, wonderfully done.
Thank you so
what
much,
content, sir. Extremely, very nicely done, very good content. Real hard work can be seen in the content which you handle. Thank you so much. And
Thank you so much, sir.
all your lectures and the PDFs are appreciated widely across. People are very happy and they really appreciate the material and the way you teach. Excellent knowledge, fantastic presentation and delivery. Brilliant.
Thank you, sir.
It's an honor to have you on this platform.
It's
4
an honor
lectures
for
back to
me.
back. Right. Great.
It's an honor. So my sincere thanks to you, sir, for
My pleasure. The pleasure is all ours.
making me a part of this lecture series of yours, sir. This is just
Yeah.
a very overwhelming. Like I don't know, I don't have words to say, sir. Thank you so much.
My pleasure. Thank you so much. Right, Dr. Shristi, thank you so much. God bless you. We will get back to you for something more, maybe in the near future.
Yes,
You
sir.
are a
Yes, sir.
brilliant teacher, let me tell you.
Thank you, sir.
Right.
Good night.